Hello and welcome to today's webinar, Whole Genome Sequencing and Analysis of ME/CFS with Dr. Camille Birch. So Dr. Birch, I'm really pleased to introduce you to Camille Birch, who will be leading that talk today. Dr. Birch is a University of Arizona graduate with a PhD in biomedical engineering. Her interests center on biophysics, cellular metabolism, energetics, genetically linked cardiovascular disease, and biomaterials, and she is driven to demystify rare and complex diseases. Dr. Birch joined Hudson Alpha Institute for Technology in early 2016, and she works as an analyst within the software development and informatics group as part of an interdisciplinary team led by Liz Worthey. The team will actually be making a move to the University of Alabama at Birmingham, where some of you may know that Jared Younger does his work. And so I think there's a lot of room for some exciting collaborations there. And so, you know, just on a personal note, I've gotten to know Camille through her involvement with the Ramsay Award program, and she's truly an incredible scientist and person. She approaches this disease with kind of an open-handed, collaborative, really smart approach. And so I'm really, really pleased that she's joining us today. And so Camille, with that, please take it away.
Okay, well, thank you very much for that introduction. So as Allison said, I work with Dr. Liz Worthey, and she was going to be here today for this webinar as well, but she had some sort of some family emergencies pop up, taking care of some stuff with the move down to UAB. But still, please ask questions about it, and we'll be happy to hear from you guys. So as Allison said, we're doing a project that's focusing on whole genome sequencing and analysis of patients with ME/CFS. So first, I'll just give you a little bit of background about us, since we are, let's see if I can make that move forward. Hold on. This was working just a second ago.
Hey, Camille, let me just make sure you've got that mouse access.
Okay.
Okay, it should work now.
All right. There we go. Okay. All right. So as Allison said, we are at the Hudson Alpha Institute for Biotechnology in Huntsville, Alabama. Just a very brief background. The founding of this institute was to use genetics and genomics to better understand human biology and disease. As she mentioned, we are going to be moving very soon, in the next couple of weeks, down to the University of Alabama at Birmingham, which is just about 2 hours south of here. That's going to be a very exciting move for us. Hudson Alpha does a lot of collaborations with UAB as well. So it was sort of a good choice for us. It opens up a lot of doors for us with expanding into more genomic medicine programs as well. And as Allison mentioned, Dr. Jared Younger is there, who's been our collaborator on this project. So we're very excited to get just physically closer, so it'll be easier to have more collaborations with him. All right, so the next part that I will talk about is just the background of who we are. Sorry, the slides seem to be a little unresponsive here. There we go, okay. So we are the Software Development and Informatics group. So as I mentioned, Dr. Worthey is the lead of our group. So just a little background on sequencing. So when you take a blood sample and you do your genomic sequencing, that sort of part of the project is sort of a field in and of itself. And this isn't really what we deal with, the sciences of doing the sequencing and optimizing the sequencing. That's what other people specialize in. But when you do that sequencing, you get a lot of data that comes off of that. And that is what our group specializes in. We specialize in taking that data, processing it the right way, and then interpreting it. And that interpretation part of the data is the development of tools, like software tools that people like myself, who are the scientists and the analysts, can actually go through genomic data. So as you can imagine, genome data is massive. There's hundreds of gigabytes of data that comes off of one person. So you have to have some sort of tool that helps you sort of digest and sort of visualize the data in a way that makes it feasible to actually do an analysis. So we've developed a tool here that's called Codicem, and this is basically just a tool that helps visualize and prioritize what you need to look at in whole genome data. So because you can see here, this is just an example. We kind of designed it to be sort of, I guess, more visually interesting. So we actually use sort of symbols and colors to help us represent the types of changes that you see from the genomic data. And Codicem has some algorithms built into it that help us prioritize what to look at first. So as you can imagine, if you're doing this and you're doing it on a daily basis, it's a lot of data and it can be really overwhelming, and you don't want to miss something that's important because you have been staring at it for 6 hours, right? So we try to develop something, and that's sort of how our group functions. Is someone like myself who has more of the biology and the science background gets to work with software developers to have that feedback loop of designing something that is not only functional but really useful for me as well. And this is sort of the first result that we've had from this iteration. So it helps us visually prioritize things. It helps narrow down the things to look at that are going to be the most likely, but it also gives us a lot of flexibility in how we look at different things. So this is just an example of like a detailed view. So if you were to click on a single variant that Cody showed you in that sort of global view for, you get a lot of detailed information from it. And this is just a simple example here. One thing that I'd like to point out is if you can see my cursor here. So this is obviously a cohort. This is actually our ME/CFS cohort that we had right now of 10 patients. And so I look at each person as an individual, but I can see what they share with other people in the context of the cohort. So we can see if things are shared or in common with other individuals, even though they're not related. We get other information like the type of change, what kind of protein change this is predicted to do, whether that protein change is damaging, et cetera. We get population frequency database information, so we can understand if this is a really rare variant or if it's more common. So we designed Cody with the idea that it needs to be really flexible. So you need to be able to filter through all these thousands of variants, right, that come out. And just as a note, the word variant, that just means essentially a mutation in genetics. It's a misspelling in your genome, and we just call them variants in the field. So you'll hear me use that word a lot. So since there's so many thousands of them that come off of an individual, we have to be able to filter that down to something that's feasible to look at. So we can filter on things like the quality of the variant, meaning does it look like it was real when it came off of the sequencer, or was it just an error? We can plug in things like gene lists of interest, so we can narrow it down on that aspect. We can look for known disease gene associations of interest, so you can ask it to filter things based on certain associations with that. You can ask it to filter things based on phenotypes association of interest, and a phenotype is just a fancy word for a symptom. So if you wanted, deafness or something like that, if you're looking for that in a patient, you could filter on that, and Cody would help prioritize the things that fit that. The other thing is you can filter it on population frequency, so you can look for things that are ultra rare or unique in that person to things that are much more common in the general population. You can look for particular types of variants. So do you want really nasty looking premature stop variants, or do you want something non-synonymous, or do you want a non-coding location? And then my favorite filter of all of these is what we call sort of the agnostic to all, which basically we'd sort of design some filters that just sort of doesn't really care about whether there's been a disease associated or not, doesn't really care what the gene is or what the change is. It's basically using other algorithms and other predictive methods to just sort of show you what's most damaging in that person. With the goal of this is to try to let the data speak for itself, essentially, and sort of help us float to the top the things that are most likely to be causing a problem, and then try to match that up to see if it makes any sense. And you can do any sort of combination of these things that I've mentioned thus far. So the point of this is just to tell you that when we do this analysis, we do it in multiple ways per person. We're trying to look for anything and everything that could potentially be interesting. So the next obvious question is, why would you do a genetic study for someone who has ME/CFS? And I would argue that there's actually a good amount of evidence out there. People have done this in the past to some degree. They've studied whether this tends to be an inherited disease. Does it have a high heritability? They've looked at the sort of a collection of polymorphisms, which are just small changes that seem to crop up in these patients. They've focused on different pathways to evaluate how genetics could be influencing certain things like inflammation. And the overall summary of these, which obviously this is just a snippet of some papers that have been put out there, is that there are interesting things to be found. However, nobody's obviously found sort of a slam dunk answer, right, for what this disease really is and whether or not it's inherited or not. But there's some interesting evidence out there to suggest that if you have the disease, someone in your family is much more likely to have it as well. So I sort of went into Liz's office one day with a whole bunch of papers and said, you know what, we do some really cool genetics work, and we do it on patients that have really rare or undiagnosed conditions. People have tried this in this disease, but I think we can apply these tools for chronic fatigue syndrome and see what we get out of it. And luckily, Liz is awesome, and she really likes us to find our own projects, and so she sort of said, go for it, and that's what led us to applying to the Ramsay Award. Okay, so before I really get into exactly what we found, I want to bring up 2 points about how diseases present in genetics, because there's sort of key things you have to keep in mind when you're looking for a genetic source of a disease. So here's an example of a protein. This is called lamin A. This is a structural protein that's sort of ubiquitously expressed across your body. It gives sort of structural stability to the cell. And as you can see, as I took a snapshot here, these are the list of diseases that are known to be associated with this one gene, lamin A. And what I want to point out to people is that one gene, one particular protein has, what is this, probably about 12 different diseases associated with it. And they range from things like a cardiomyopathy disease, Charcot-Marie-Tooth disease, which is a neurodegenerative process, muscular dystrophies, lipodystrophies. So you have a broad range of diseases that by themselves don't really seem connected, but are all caused by a problem in this one gene, lamin A. And you can also see the mode of inheritance highlighted over here is different for these diseases. Some of them are dominant, some of them are recessive, some of them actually have both forms. And essentially this disease is determined by where in the protein the problem is occurring. So where it's occurring is going to determine whether you get a cardiomyopathy type phenotype or a muscular dystrophy or a neurodegenerative process. So this is important to keep in mind. Now here's the opposite situation. There's a great disease that's sort of the poster child for many different subtypes called glycogen storage diseases. There are 14 known subtypes for this disease. What's interesting about this is it's the opposite of lamin A. Each one of these subtypes is caused by a defect in a different gene, but all these genes are linked because they're all involved in either glycogen synthesis, breakdown, or glycolysis or glucose breakdown. So it's all how your body handles glucose and its constituent parts. And these subtypes range from really, really early infantile onset, very severe diseases that can be lethal if they're not treated quickly, to much more mild forms that are adult onset. So again, it's the same family of disease, but it's caused by different genes being affected in that pathway. So when I approach trying to do a genetic study for chronic fatigue syndrome, ME/CFS, this is what I have in my mind, is that we don't know what the etiology of this disease is. It could be a situation where it is one protein, and depending on where the problem is, gives you a different presentation. Or it could be many different proteins that are having a problem, or different genes, but they're related by the pathway that they're involved in. Or it can kind of be somewhere in between, right? There's multiple ways that diseases can present with a genetic origin to them. All right, so the cohort that we have worked with, we had 10 patients, and we were able to work with Dr. Younger to select patients from his cohort. And these patients, excuse me, had gone through pretty extensive prior screening through his group to qualify as an unexplained case of pain and fatigue. 8 out of 10 of these patients actually had been given a clinical diagnosis of one of these terms, right? ME/CFS, systemic exertion intolerance disease, or even fibromyalgia. Most of them had both. Most of them had an ME/CFS/fibromyalgia diagnosis. Now, it's important to note for those 2 patients that we didn't actually have a clinical diagnosis, one actually noted that they were so sick they couldn't have any insurance, and so they had no medical care basically. And the other person did not provide any info. So I still take that as a likely ME/CFS case, right? They don't need to have necessarily a clinical diagnosis. They obviously are very sick and they've made it through Dr. Younger's screening process. So we know it's not some other obvious problem like a thyroid problem or a major infection going on. So these patients give a lot of information to Jarrett when they sign up for his research protocol. We get information off of them like self-ranked daily pain and fatigue. There's some detail to that, like whether the patient reports it as widespread or limited to specific regions. We get other diagnoses that they might have. It's very common in these patients. I think we can all agree IBS, migraines, things like that are quite typical to see along with ME/CFS. We get their list of current prescriptions that they're on, what their occupation is, if they can hold an occupation. We get general blood work values on them, and then a survey of symptoms that Dr. Younger's group developed. And this asks sort of specific questions like, when did your problems start? How long did it take to develop? Do you feel like you're getting worse? Are you getting better? Or are you kind of stable? Now, there was also an opportunity in this survey that they fill out for the patients to sort of write a descriptive narrative of their illness and their progression and the symptoms that they have. And as I will get into a little bit later on, this was really, really helpful for us when we were doing the genetics analysis for those patients who did add this extra layer of detail to it. And I'll get to why that's so important in a couple slides here. So this is just an overview of who we're working with for this very first pilot study. So the first thing I'm going to talk about with our results that we have so far is something called a risk loci. So a loci is just the name for a location. We have to have fancy words for how we describe these things. And a risk loci is typically thought in genetics as something that by itself does not necessarily cause disease, but it is it makes you more susceptible to developing a disease, right? So there can be certain risk loci for developing cardiovascular disease, right? It doesn't necessarily mean that you're going to have that, but you're at a higher risk for developing it. So when we did the analysis on these patients, let me see if my bullets don't want to come up. I think sometimes there's a little bit of lag here. Let's see, I don't know why. Did I lose control, Allison? Because these bullets are not popping up. Nothing's progressing.
It looks like we're having a little bit of a lag, so let me just troubleshoot that.
Okay. Oh, there we go. Ooh, here we go. Okay. So what we started by doing with these samples was we flagged some of the variants that are more common in the general population, but they had a known functional effect. So these variants had been studied. They were known to affect how the protein worked. They were just, and they certainly weren't rare. They were in, 10%, 20%, 30% of the general population. Now, many of these, I can tell you, were in this family of proteins called the interleukin protein family. And these are immune proteins. They're cytokines, either receptors for them or the cytokines themselves, which I thought was pretty interesting. And what we noted was that we only have a small number. We have 10 patients, but we kind of saw that some of these, although they are more common variants, they seem to be kind of at a higher than expected frequency in our little cohort. So we asked the question was, could we rank how probable it is if you were to sort of randomly select 10 people, how probable would it be to see this in 8 out of 10 or 9 out of 10 when it's generally in 10% of the population? And if this came out to be highly unlikely, it may, and I very much stress may because it's a very small number we're working with, indicate that this could be sort of like a genetic signal that means you're more susceptible to developing this type of disease. Now, obviously, the larger your cohort, the more certain of this type of association you will become. But it's hard to do statistics on really tiny numbers like this. So sort of doing this kind of probability was sort of the best thing we could do with a small number. Okay, so overall, we had 32 risk loci that we identified in these 10 patients. We perform this thing called a binomial probability, and essentially it asks this question down here at this bottom bullet: if 30% of a healthy or ostensibly healthy population database has this variant, what's the probability of finding it in 8 out of 10 patients? And when we did this, it resulted in 5 of those 32 loci being very, very unlikely to be present in that number of the population that we have, given that known population frequency. And so that's what we have here. So this is a little busy, but I'm just going to walk you through what we found really quick. So you can see in the column here, population frequency. This is what it is in the general population. So this one gene, AMPD3, this variant's in about 6%, 25 to 30% for the others, and another at about 6% of the population. So definitely not rare variants. Obviously, in a lot of people that don't have ME/CFS, because it's not quite that prevalent. But what we found was then some of our patients, for instance, the AMPD3 gene, this was in half of our population. And some of these were in 7 out of 10, 8 out of 10, or 4 out of 10. And so when we ran this calculation to say, well, what's the probability of seeing that if you were to randomly select 10 people, these were the ones that came out as less than 8% of a chance of seeing this. So AMPD3 was particularly rare. This is already converted to a percent. So this is about 1/100th of a percent chance of sort of randomly finding this. Now, exactly what this means is sort of still up for debate. I'm not quite sure, but the function of this is pretty interesting. So AMPD3 encodes an enzyme that basically helps catalyze the change of adenosine monophosphate to inosine monophosphate. So this is part of that energy regulation network. I'm sure a lot of patients are familiar with what ATP is, or adenosine triphosphate. So this is sort of the breakdown products of that, and it's how your cell sort of regulates the energy currency that it has. So this was kind of an interesting thing, because there is actually a known deficiency of this enzyme, but it's not supposed to be clinically actionable, meaning it doesn't really present with any symptoms. So this actually may be a good susceptibility candidate for developing a problem later on. You can, you can see that the other 3 were these interleukin proteins. Like I mentioned before, these are immune proteins. They're either cytokines that are produced by particular immune cells, or they are the receptors for those cytokines. So they help the immune cells talk to each other, essentially, and they help drive inflammation. The last one was kind of interesting, which is this NOS2 gene, and this encodes nitric oxide synthase. So this is an enzyme that helps your body produce nitric oxide. And nitric oxide is one of those molecules in the body that has an incredibly wide range of activities. It's a very potent vasodilator. It's also a very potent signal to modulate inflammation. So overall, these were the 5 that had very, very small percent chances, you could say, of showing up in these 10, or in this number of patients that we have, given what we know the frequency in the population is. So it's a little bit tough to know exactly what this means right now, but it's an interesting first step, and it's an interesting finding to have in just this very first study that we're doing. All right, so now I'm going to talk about what we found that were the rare or the unique variants in these patients, and this is really the area that we specialize in. So on average, each patient had about 14 rare or unique variants that passed our analysis and our team discussion. So this was basically our short list that came out of all these different types of analyses that we did, and discussion as a group as the most interesting things that came from these patients. So again, just emphasizing, each one of these patients was analyzed sort of individually, and they were analyzed in multiple ways. So this included doing different gene lists. We were looking for different frequencies of things and different damaging predictions. So we really tried to cover our bases as best as we can, because we don't know what the source of this disease is, and we wanted to just sort of slice and dice the data multiple ways to make sure we weren't going to miss something obvious. So what I can tell you is what we found was that they hit a range of pathways. There were lots of metabolic things, immune things, there were several ion channel things, and some nuclear encoded mitochondrial things. I can say, because we did a whole genome, that we also get the mitochondrial genome as well. And we didn't see any mitochondrial genome variants. These are the variants that are encoded by your nuclear genome that can affect the mitochondria. So what's the difficulty with this? Well, What was hard was that when we have 10 patients, most of these variants were unique to each patient. So the difficulty is finding the common thread, right? How do I know out of these, on average 14 per patient, which one, if any, could be responsible for their disease? Clearly something is wrong with these patients if they've gone through all that effort to be a part of this research study. But that's where the challenge really lies with genetics. And this is where I'm going to take a small detour to tell you why getting these phenotypes on these patients is so critical to what we're doing. And I think really critical to advancing our knowledge of this disease. So why are phenotypes so important? So I actually, when I was doing this analysis, I realized that on the Solve ME/CFS website, there's a tab there called the Humans of ME/CFS, right? Many of the patients will be familiar with this. And these are just stories that people voluntarily tell. They upload their story about what it's like to live with their disease. And so as I was going through the analysis, I thought, man, I really need to know more about how this disease presents. I feel like I have a good understanding of this. As I think some of you may know or have heard me talk about before, this disease does run in my family. And so I feel like I'm pretty familiar with it. But as I was doing this analysis, I realized, maybe I'm not as familiar as I thought I was, or maybe there's a wider spread of how this disease presents than what I might initially think. So since this is sort of freely available, I went online to these stories and I started reading them, and I started taking notes of what people would describe. Now, obviously not everyone would describe in detail how their disease presented, but actually a lot of people did. And I got through about 60 stories, and I would like to continue to do this because that's a little less than half of what's on there. And I'm going to tell you what I saw and what I noticed initially from that. So again, my goal was to try to categorize how these patients' disease, what was the onset of their disease, how old were they, and did they mention any really particular symptoms as standing out as one of their, the most difficult things to deal with the disease. So what I found was that about a third of these patients that I read described this relatively sudden, and I think a lot of us know this story, relatively sudden, meaning days to week onset that was very much initiated by an illness. Most of the time it seemed to be the flu or some sort of type of flu. Some of these patients actually had confirmed cases of mono or Epstein-Barr or other infections like meningitis. And the vast majority of these stories that describe this type of onset were adults. So it was typically in their late 20s onwards, and there were some that were even as late as their 50s or 60s that didn't experience that onset until then. About 10% of the stories really indicated that they were never normal. They would describe a picture where they recalled having problems and feeling different from their peers, even when they were kids, but that the problem didn't get really bad until they turned through puberty, basically, till they hit their teen years to young adult years. And they always seem to describe this more gradual, slow decline. It might take some step function decreases, but it was never very sudden. It was sort of always present and just sort of continued to get worse through time. Now, there was another small group of patients that I decided to sort of bin separately because I'm not quite sure if this could be something different or not, that described what I call a rapid onset. And by rapid, I mean within a day, that was triggered by some sort of event. And this means something like a surgery that happened after some sort of trauma, like a car wreck. Some people described it happening after like a medical procedure, like a transfusion, or sometimes even like a really severe stressful event, like an unexpected death in the family. And I decided to distinguish these from the other two because what they described was a drop-off in health that was absolutely massive and was really rapid, usually over a 24-hour period. And an illness did not appear to be playing a role. There was nothing going on infection-wise or some sort of flu-like symptom. It just sort of hit them quite literally out of nowhere one day. And in addition, there were a lot of other detailed symptoms that these patients would describe that seemed to help bend people into different buckets, essentially, including things like having really severe cognitive problems, which was something I was actually kind of surprised by. I know and certainly have experienced those cognitive problems, the cognitive fog, having difficulty finding words, but there were a few people that would describe something absolutely so severe. It sounded almost like what you would hear with something like Parkinson's, right? They couldn't even follow a sentence. They couldn't even follow a plot of something. Just really, really bad cognitive problems that go beyond just that kind of what you sort of hear as like the cognitive fog feeling. A lot of people, but not all of them, which I think is important, described really bad fibromyalgia type symptoms, and some also described like really severe orthostatic intolerance problems. And so the reason I bring this up is that each one of these situations that I described to you could very well, from a molecular point of view, which is what I'm trying to view it as, be something different, right? And so I needed to get a feel for what was the spread of stories and the spread of symptoms going on in this disease to sort of help me understand the patients that we're working with, and what kind of questions do I need to ask of them to sort of understand them better, so that I can ultimately understand what their genetic result means better. So I say this because phenotypes, right, or symptoms, they're the warning signal. They're the signal from the body that something's wrong, right? Now here's something about genetics that may not be as obvious, which is when you have someone's whole genome, if you were just given a whole genome from a person, you actually don't know an awful lot about that person. You know a good amount because it's a lot of data, but you don't know everything by any means. So there are things that are real obvious. You could probably guess, if you knew nothing about that person, you could guess maybe their ancestry. You could guess what their eye color was. You could find things that they might be a carrier for, like cystic fibrosis. You could find things that they might be at risk for, like those known cancer risk variants. But what you can't guess is if they're is what disease they are presenting with right now, if any. That's actually really difficult to do with no other information other than the genome. So that's why I say the type of genetics that we do relies really heavily on getting an accurate clinical picture or an accurate phenotype or symptom description. Those are all sort of the same thing. And I say this because recall those 14 different types of a glycogen storage disease. That's 14 different subtypes caused by the same pathway being hit in different ways, and they present in different ways, different ages of onset, different severities, and they will respond to different treatments. Excuse me just a second. Oh, terrible time for allergies right now. Okay. So the clinical picture from these patients is what's going to be really key to helping us identify what is most likely going to be the molecular or genetic cause. Those are kind of the sort of interchangeable words for this disease. And this is what my goal is. I want to help figure out what is going on with all these patients with these mysterious symptoms. And you can only go so far with just genetic data. You really need that extra bit to really kind of push you over the edge to sort of find the most likely candidates. So I also want to emphasize that when we do this analysis, each person in our analysis is important as their own. They are important as their own standalone case and in the context of the group. And what I mean by that is we try to approach this where we analyze each person as their own case, their own individual, and whatever I learn from that individual, I can then apply to the group, apply to the problem that I'm trying to solve. So what did we learn from these 10 patients after I've gone down that little bit of a divergent there? Well, we learned that we needed to know a bit more about them in order to sort of take that final list of 14 or so interesting things down to maybe just one or two. So what I did was I was able to write an additional set of questions. And as we're working with Jared and his group, these are his patients. And so they're able to recontact them, which is excellent. So we were able to send this out. And additionally, with not only some extra, I guess, finer details that I was asking for on how their symptoms present, I also had some spaces in there where I kind of asked them to just give a descriptive narrative, like in their own words. Because for me, that's very helpful to sort of gauge how severe someone is and what their daily life is like, which just sort of helps put in perspective the other questions that they're answering, yes or no on, for how they experience things. Now, despite this, we still did find some really interesting things in each person, even though we've just gotten some of the questionnaire data back, and I've just started to look at it. But I'm going to tell you what we found so far in these 10 patients as the interesting things. And there's going to be a recurring question that comes up with each one of these categories, but I still think this is really an interesting start. So the first category I'm going to talk to you about is energy metabolism. Now, what I mean by energy metabolism is essentially on a cellular level, how do you regulate how your cell uses energy, right? And particularly things like ATP, how does it regulate its energy currency? This is obviously an area that I really like. This is sort of what I did for a while in grad school. And surprisingly, I actually found some pretty interesting things in this. Now, so here they are. So there are 3 patients here. Each one of these is different. So patient 910, 903, and 905. And each one of these had a variant that looked quite damaging, and as you can see by the population frequency, is extremely rare, very, very rare. And what's important to note is that each one of these genes is in a related pathway. They're all part of this, it's called this AMPK energy sensing pathway. So many of these energy regulation genes actually have never been associated with a disease before. And so that's also sort of interesting from a discovery standpoint. It could be that these actually might be causing a disease that we just haven't described yet. And so what I thought was unique was that these 3 patients these 3 genes sort of interrelated function. So this one gene, PRKAG1, is actually sort of the energy production gene, so to speak. It sort of tells your cells to ramp up energy production. Genes like mTOR here is actually the energy brake gene. It tells AMPK to quiet down and to use energy. And then ULK1 is sort of like a mediator between those two. So these are actually really tightly related from a molecular point of view. And I thought that was kind of interesting for these to come up in these 3 patients. So from a molecular perspective of how that could work, the role could make sense to alter some sort of fundamental or basic aspect of cellular energy production or regulation. And maybe that's what's driving some sort of disease mechanism. So the question comes up, which comes up in each one of these categories I'm going to talk about, is could this be explaining a portion of these patients? Now, that's hard to say right now, as I hope I was able to show to you with needing that extra clinical data, because I'm not quite sure exactly how similar to a very fine level these 3 patients are. As we move forward, if these 3 patients have really similar stories, this might bring these up to our attention where we need to sort of further investigate them. We actually need to do some sort of like bench study work to see if this could be actually affecting function in some way. So the next category is iron metabolism, and this was a bit of an unexpected one. But what we found was there were 2 patients. So there was 1 in patient 908 and 2 in patient 907. We found damaging rare variants in iron metabolism genes. So this first patient had this gene TF. This is transferrin. So this is the protein that actually helps you transport iron, particularly from your intestines. It helps you absorb it from your food and sort of deliver it to all your cells. This TFRC gene is actually the transferrin receptor gene. So this is sort of what binds the transferrin once it's in your bloodstream and helps deliver it to that cell. And then this HPX is called hemopoietin, and this is a protein that is particularly highly expressed in your liver that actually helps you recover iron through breaking down your old heme molecules. And again, as you can see, these were really rare. These last 2 were not even in the population frequency database, so it means that they're probably unique to patient 907. And they were all predicted to be damaging. So this brought up another interesting question, which is like, well, could these iron metabolism genes actually be playing a role for some of these patients? If anyone out there has ever experienced anemia of any type from any sort of cause, you know that anemia makes you feel absolutely terrible. I mean, it is a system-wide problem that you can have. And what I thought was interesting was if you look at some of the animal model data on looking at how these genes function, most of the time they don't lead to blood anemia. By blood anemia, I mean your blood values would still look normal, but it's quite likely, what they found was that tissue anemia was actually a problem. So you can get the iron in your blood, your hematocrit is fine, your red blood cells can bind iron just fine, but it doesn't get delivered to your other cells like it should be. And so you might have an issue of tissue anemia, which is obviously much harder to diagnose, that could be driving a problem. And again, the question comes back to, maybe this is a subset that's having a problem with this. I'm not quite sure yet. It's going to take more investigation and, again, more patients to see if some of this stuff starts lining up. Now, the last one is something I actually brought up before, and if iron metabolism was not on my radar, I can absolutely tell you that glycogen metabolism really wasn't on my radar whatsoever. But we found two pretty interesting things, and again, two different patients out of this group in this pathway. In particular, this patient 911 had a gene called ENO3, and this encodes an enzyme called enolase 3. It is in the glycogen metabolism pathway. It's very highly expressed in skeletal muscle and to a lesser extent in your heart. And what's interesting is there is actually a known disease associated with this gene, and it's called glycogen storage disease, or GSD type 13. And like I mentioned before, a lot of GSDs are really young onset or infantile onset and usually cause seizures and extremely severe phenotypes. This one is known to be adult onset. Typically doesn't happen until you're 30 or older, and it's a much, much more mild form. And the primary symptoms are severe muscle pain, especially after any sort of exertion, and a near complete intolerance to exercise. Now, this next patient, 904, also had something in this glycogen breakdown pathway. However, this gene, GAN-C, has actually never been linked to a disease. It's just we know that it is involved in that starch and sucrose metabolism and the glycogen breakdown. So this was a pretty interesting finding because what you can actually do, unlike the other two that I talked about, this is actually a little bit easier to go confirm if there's actually something going on here in these patients. You can actually do a muscle biopsy, which is a slightly invasive procedure, but it can be done. And you can go look for glycogen metabolism or glycogen storage problem in the muscle. And that's a slam dunk way to diagnose whether this problem is existing or not. So we're going to see if we can follow up with this patient because we're quite convinced from the genetic data, at least, that this could be causing a problem for her. She was also a patient that was a little bit more mild than some of the other ones that we had, which kind of fits with the description of the symptoms that are associated with this type of GSD. And I want to note that this type 13 is thought to be very, very rare. I think there's only 2 families that have been described in the literature that have been found to have an enolase 3 deficiency. So this brought up an interesting question in our group where we've seen this happen in genetics before, where we think something is extremely rare, but it's actually just really hard to diagnose. And if you actually knew to go looking for it, it might actually be slightly more common than what we thought, right? So this brings up an interesting question of maybe there's a more mild form of an adult onset, perhaps caused by this ENO3 gene or maybe this GAN-C gene, that could be more common than we think and could actually be easily mistaken for having something chronic fatigue. And so overall, I just want to point out how, we all know that there's a problem in this field of getting diagnosed with this disease. And I think it is so easy for someone to just be labeled with chronic fatigue syndrome because the physician or the specialist just doesn't know what else to do with the patient. They don't know what else other tests to run. And what I hope we can do with this work is try to untangle this a little bit, right? Maybe there's a portion of patients in here that are just misdiagnosed. And then maybe there's another portion that have a brand new disease mechanism. That's completely possible. And what's going to be key to that as we do this work is getting those really detailed stories, looking at each person sort of as their own case, and then trying to apply that to the general cohort and whatever we learn from that person. So what are our conclusions overall? Okay, so we found some unexpected and what we thought were quite interesting findings in just these 10 people. The most obvious pathways that popped out were these basic cellular energy regulators, iron uptake, transportation and salvage, and glycogen metabolism. And I want to point out that the patients that had variants in this cellular energy metabolism didn't have anything in iron or glycogen, and someone with iron didn't have anything in the cellular or glycogen, or vice versa. So these were really the unique pathways that popped in these patients. It's not like we saw that in all of them and sort of only pulled out the ones that were interesting. So our next steps for this is that we would like to, once we kind of get settled at UAB, since we have our transitioning happening now, we're going to circle back to some of these and see if we can pursue some molecular or some sort of laboratory confirmation on those that we find the most interesting. That might be possible for some of this. Obviously, as we move forward with this work, we'll get more patients, which means more data, and we'll get more detailed and careful phenotyping that will hopefully help untangle some of these questions that we have. It's quite possible that some of these avenues we think are interesting right now may not pan out. They may not be what's actually causing a problem, but we'll know that the further we go with the more patients and the more data that we get. And so something that Liz and I really want to emphasize, especially when we work with other collaborators, is we're really trying to use our skills are in this area, genetics and informatics. And we want to use this to help lay a foundation of data so that we can share it with others so that we can better understand this disease. Or as I said, maybe diseases, maybe there's more than one thing going on here with these patients. And that's really what our interest is. And I really love working for Liz because she's a very collaborative person who really believes in data sharing and having good collaborations. So the future work, we're going to get more patients and more data. I have to give a very big thanks to Jared right now for helping us gain access to these patients. If we didn't have that collaboration, it would be really hard for us to get these samples. So he's been absolutely wonderful to work with and to share our data with him of what we're finding. And right now, we have had some talks with some other research groups in the ME/CFS world and those outside of that world, more in the genetics world that we live in, to help expand these studies. And I can say, I was mentioning this to Allison right before the webinar started, Liz was able to bring some of this work to a clinical medical genetics conference that we went to back in April. And these are a lot of doctors and physicians that go to this conference who are geneticists. And it was received really well. There's a lot of physicians out there who look at this and say, God, I really wish somebody would help me answer this because I don't know what to do with these patients. And I think there's a bit of a change in the physician's mindset now. They feel stuck. They feel like they don't quite know what to do, how to treat them, but they're really happy to see folks like us sort of show up at these conferences and talk about the work that we're doing because they're very eager to understand it as well. So we also have a little bit of data that we're finishing right now. We're getting some RNA data on these 10 patients. So this is sort of the expression data for what proteins are being expressed. And we also have just wrapped up some immune repertoire sequencing right now. So hopefully we can get sort of an immune profile from a genetic point of view on these patients. And we'll have to learn how to interpret that and then pair it up with the whole genome data that we have at the moment. So that's the end, basically. There are a lot of people to thank for this. I certainly don't do any of this work in a vacuum. Our group has been fantastic to work with this. As I said, Jared is an excellent collaborator. He's been very open to talking with us and allowing us to sort of piggyback off of his cohorts that he's bringing in to get some of these samples in. And obviously a big thanks to the Solve ME/CFS initiative for sort of funding a crazy idea, I guess. And we're very, very excited to do this work. And a huge thanks to any of the patients and their families who are participating in this, because I certainly know that that is not easy to fit that into your life, especially when you're very ill. So thank you very much for listening, and that's it.
Hey, Camille. Thank you so much for such a great presentation. And we were really thrilled to fund this work. You know, the sort of combination of an unbiased approach and deep dive into the literature, the really informed perspective that you take on the disease is really a powerful combination. And so, I mean, we're really excited to see the rest of your study play out. And there were definitely some questions coming in. The audience is engaged. And so I think I'm just going to kick it off first just to jump off from your point that some of the next steps for your team will be to get more patients included in this study. We had a couple questions come in about whether or not people can donate their whole genome data to you, and if there would be any kind of protocol in the future for you to incorporate, you know, data sets that already exist into your project.
Yes, that's an excellent question. We get asked this quite a lot, and the short answer is yes. Since we have this transition, we've been sort of, anybody who's moved anywhere knows it's crazy, right? And we're moving an entire lab. So it's been a little bit, everything's been on pause at the moment. But when we get to UAB, this is absolutely something that Liz wants to set up, whether it's, I know like SMCI, you guys are building biobanks and things like that, whether we do it through some sort of source like that, or we set something up at UAB. The short, again, it's yes, we're really interested in getting stuff like that because that's so easy. You don't have to give a blood sample, the data's already there. You just give us the data and we can process it and interpret it. So I guess keep your eyes peeled because we certainly will be wanting to do this in the next couple months over the summer, for sure.
That's great. That's so exciting. It's definitely a goal that we're really aligned with. And another participant mentioned that Nancy Klimas's group at Nova Southeastern University recently published a paper that included data from people's 23andMe, WGS data. And so you mentioned that you've been doing a lot of proactive outreach to some groups, both within the field and genetics experts that aren't currently working on the disease, to see how everyone might collaborate a bit better. So can you tell us a bit more about how you guys are trying to stoke collaboration, whether you're working with any international collaborators, and how you think some of these efforts can, be synthesized together a bit?
Yeah, that's a great question as well. So I was able to meet Nancy at the conference that just happened at NIH, and she's doing fabulous work as well. 23andMe data is a really great, quick and easy resource, right? Because it's it's just a saliva sample. You don't have to do any blood draw. The patient can do it from home. It is a panel, so it's essentially a snippet of the genome essentially, but it's a great place to start. And I was so happy to hear somebody was approaching that problem. So people like Nancy, absolutely, we're ready to collaborate for sure. And we've also had some other conversations. Liz just had a conference over in the UK just a couple of weeks ago. And she actually met the fellow doing the UK ME-CFS research there, and he sounded incredibly interested to work with us. I know they're doing some really big GWAS studies, so these are like the really big statistical analysis studies on genome data. So they have to recruit lots of patients, hundreds of patients, and he was very interested to work with us where maybe we could take a number of those patients and do a whole genome sequencing and then pair it up with what they're getting off of their statistical analysis. So that's definitely something we're going to follow up with over the summer. Excuse me. That's sort of the number that we're working with right now. And then working with Jared has been fantastic. That's going to continue to move forward as we get more data and write some papers off of this initial stuff that we're looking at right now.
That's great. It's really, as we've been working at Solve to try and better integrate some of the folks that have received, Ramsay awards together and to form kind of a network of people that can better share data and collaborate. And all of the work that you and Liz have been doing to do outreach, most importantly, even externally to the field is just really, I think it's really inspirational and exciting. And there was also a couple of folks that kind of had a question about the immediate potential translational benefit of this type of work. As there's a lot of people that are really, really sick now, and there's a real drive to get something that can offer relief as soon as possible. And I think the methodical approach of defining subsets and peeling them away to help try and identify targets that might work for groups of people is one of the most promising approaches to solving ME-CFS or this kind of heterogeneous group of folks. And so, is there anything immediately available for people that they could try? You mentioned a really rare, type of disease that could potentially explain some of the symptoms that people are experiencing. But, just even if you could offer a sort of broad look of what you think the power of defining subtypes might look like and how that could unfold, I think folks would be interested to hear that.
Yes, absolutely. So I can tell you, so personally, my view on this problem is I could spend the next 30 years trying to figure out and understand all the intricacies of what's going on with this disease. This is what we're doing with all these other diseases, right? Cancer, for example. We know a lot about it, but we certainly don't know how to fix it for everybody. But there's a lot of options for people with cancer, right? And every year, every decade, it gets better and better. So with this disease, what I see is sort of a short-term goal. It's sort of like my couple-year, five-year goal, which is to really help define who these patients are. It's so hard to get diagnosed with just fatigue. I mean, that doesn't tell you anything. And if you can define that and you can start showing, look, 30% of these patients, we've done X number of studies, 30% tend to have this molecular problem. Either you can start repurposing drugs that already exist, or you can at the very least start getting the attention of pharmaceutical companies, small or big, to start working with you because you have the numbers now to back up what you think the actual mechanism is to start actually trying to treat these patients. I know hands down, that's what I want to do with this work, because it's not really much good to understand it without knowing how to try to fix it, right? So we'll spend a long time trying to understand all the intricacies, I'm sure, but I think that in reality, we probably can do quite a lot for the next, 5 years to try to get something going where this is either more recognized as a problem of diagnosing these patients, of misdiagnosing a certain number of them, or having a brand new cohort or a brand new disease mechanism found that you can get a company involved to sort of start developing therapeutics. The pharmaceutical industry is never going to touch this if they have no idea what to even aim for. So, which of course they wouldn't. That doesn't make sense for them. So you have to, I feel like it's our job, our work, to try to lay that foundation so that that work can actually progress forward. So I hope that answers your question.
Yeah, absolutely it does. And I think it's kind of, we're getting close to the top of the hour. I think that's an inspirational note to end on. I know there's a lot of really great work going on in the field, from the group at Stanford to the work that you and Liz are doing with Nancy Klimas. And I think some of this work is driving really interesting aspects of the research that has a lot of promise to produce some really tangible answers in the near future. And so thank you so much for doing this presentation today, and for being part of the group of Ramsay investigators that are currently working. We really appreciate it.
Well, thank you very much. I was very, very happy to share what we have so far.
Great, thanks, Camille. And I'm just going to give folks a reminder that we are going to be hosting this webinar series throughout the year. You can find more info at the link. We're still scheduling out some future webinars, but we look forward to bringing them to you soon. If you want to stay informed, you can do so at solvecfs.org/newsletters. And of course, if you're able to give, that really helps fuel the work and helps us fund more researchers through our Ramsay program like Camille. So thank you again to everyone for joining, and yep, thank you very much. Bye.